Genetic transmission in viruses. This is a microbial genetics portion. In living organisms, the genetic material is well packaged and is transmitted from parent to progeny in a highly ordered and precisely programmed manner. In multicellular organisms, genetic transmission is of two kinds, lateral and vertical. Lateral transmission always occurs from parent cells to daughter cells, whereas vertical transmission occurs from parent organisms to daughter organisms. In unicellular organisms, there is no such distinction because full-fledged organisms are only glorified cells. The genetic information inherited by an organism is phenotypically expressed through the synthesis of specific proteins. In sexual and pansexual mechanisms, genes are transferred from parent to progeny as reshuffled packets. This is mainly due to an orderly programmed process of gene shuffling known as genetic recombination. Recombination ensures that each new progeny will have a particular gene combination a little different from parental combinations. Yet it retains the species specific traits since it conserves the minimal quota of the basic gene combinations and the basic traits of the species. The mechanisms of genetic transmission are different in viruses, bacteria, and eukaryotes. First, viral genome and genetic transmission in viruses. Viruses are infectious intracellular obligate parasites with subcellular level of organization. They are devoid of protoplasm, cell organelles, enzymes, and the molecular machineries for energy metabolism and protein synthesis. They are extremely simple, ultramicroscopic organisms formed of only viral proteins and DNA or RNA. They lack the powers for metabolism, so they depend on the host cell for the raw materials and enzymes for essential functions. These viruses can multiply and undergo mutation only in living host cells. Outside the host cells, they are simply inert particles. First, bacteriophages. Bacteriophages or bacterial viruses are the viruses which parasitize bacteria. They were discovered by the British scientist Todt in 1915 and later described in detail by the French scientist De Herelle in 1917. They are widely distributed in nature and are especially found in large abundance in enteric bacteria which inhabit the intestine of man and other animals. They are especially adapted for adaptation and penetration through the rigid cell wall of bacteria. There are several varieties of phages. Usually each variety is specific to a particular species or strain of bacteria. Bacteriophages fall under four major groups, namely single-stranded DNA phages, double-stranded DNA phages, single-stranded RNA phages, and double-stranded RNA phages. And we try to learn about the typical structure of a DNA phage. T4 bacteriophage, commonly called T4 coliphage, can be taken as a typical bacteriophage. It is double-stranded DNA phage with binary, that means cubical and helical symmetry. It parasitizes the intestinal bacterium Escherichia coli, hence the name T4. Hence the name T4 bacteriophage. It has five structural parts, namely head, neck, or head to tail connector, tail, base plate, and tail fibers. Head is shaped like an icosahedron. It is an elongated bipyramidal and hexagonal prism which is about 95 nm long and 65 nm in diameter. The central core of the head is formed of a linear and double-stranded DNA molecule associated with some cations, several peptide, and three internal proteins. The DNA is characteristic in having hydroxymethyl cytosine instead of cytosine. The core DNA is encased by a capsid formed of about 2000 capsomes or protein subunits. Head-tail connector or neck is a very short region in between the head and neck. It consists of a collar or disc and several fiber-like whiskers. Tail is a narrow cylindrical part about 80 nm long and 18 nm wide. It has a central helical and hollow core or tube through which the viral DNA passes to the host cell during infection. The core is surrounded by a helical and contractile sheath of proteins. It is formed of 144 protein subunits arranged in 24 rings of 6 subunits each. The sheath is connected to the collar basally and to the base plate terminally. Base plate or end plate is a hexagonal plate with a pin or spike at each corner. It is terminally connected to the tail fibers. Tail fibers are 6 in number, arising from the 6 corners of base plate. They are used for attachment to the host cell wall during infection. Each of them is nearly 130 nm long and 2 nm wide. Each fiber has two portions or half fibers, proximal and terminal. Proximal half fiber is attached to the base plate, and the terminal one is used for attachment to a receptor site in the cell wall of the host. So tail fiber have the power to recognize specific receptor sites in the cell wall of the host. So regions like head is shaped like icosahedron symmetry, then whisker, collar, hollow neck from head to base plate, tail or helical symmetry tail, then tail fibers, spike hexagonal base plate, etc. are present. Then viral genome. Viral genome is extremely diverse. It consists of a single-stranded or double-stranded RNA or DNA. Viral genome may be composed of one of the following: single-stranded RNA chain, double-stranded RNA helix, single-stranded and linear DNA, single-stranded and circular DNA, double-stranded and linear DNA, double-stranded and circular DNA, and double-stranded DNA with covalently bound terminal proteins. Since viruses are able to replicate only inside their host cells, viral genome must be organized in such a way that it may be easily recognized by the host cell. So also the viral genetic code must be matched to that of host organism, and the control signal of gene expression must be somewhat similar in both. Viral genome is very small with only much limited store of genetic information, so it can code only for a few proteins. Consequently, it cannot replicate and phenotypically express all by itself for want of necessary enzymes and protein factors. Therefore, a virus depends upon the molecular mechanisms and cellular machinery of its host for genetic transmission and expression. To accomplish this, viruses take control of the molecular machinery of the host cell to synthesize their nucleic acid and coat proteins, and also for the formation of their capsid. DNA viruses and RNA viruses employ different mechanisms for gene transfer and gene expression. Then genetic transmission in DNA viruses. The mechanism of gene transfer is different in different kinds of DNA viruses. Based on this difference, DNA viruses are classified into lytic viruses, lysogenic viruses, and tumor-causing tumorogenic viruses. Lytic virus produce many progeny viruses in the host cell and ultimately cause the lysis or disintegration of the host cell. So they are also called virulent viruses. Lysogenic viruses live inside the host cell without producing viral particles and without causing the lysis of the host cell. So they are also called non-virulent or temperate virus. Most lysogenic viruses undergo lytic cycle also under certain circumstances. So they have a dual method of genetic transmission. Through an open strategy of lytic cycle, they produce the maximum forcible copies of viral genome as rapidly as possible. Through an alternative closed strategy of lysogenic cycle, they integrate the viral genome with the host cell genome and inherit it as a part of host genome. Tumor-causing viruses abnormally alter the physiology and morphology of the host cell, make the cell cancerous, and thereby induce tumor formation in multicellular organisms. T4 and T7 bacteriophages, which infect the bacterium Escherichia coli, are lytic viruses. Lambda phage, which also infect E. coli, is a lysogenic virus. Rose sarcoma virus or RSV found in chicken is a tumor-causing virus. The method of gene transfer in phages is determined by the nature of the transcription regulation of the phage genes. In lysogenic transmission, phage genes are repressed by the interaction between a phage repressor and phage operator. In the lytic method, there is a cascade of transcriptional controls in which successive sets of genes are successively turned on at specific times. Each set contains a regulator gene that brings about the transcription of the next set. Then gene transfer in lytic viruses or virulent viruses. Gene transfer in the T4 lytic phage is an extensively studied phenomenon. In this case, the virus first absorbs to specific surface receptors on the bacterial cell wall with the help of tail fibers. At the point of this attachment, the bacterial cell wall dissolves and an opening is formed by the action of viral genome lysoside. Through this opening, the viral DNA is injected into the bacterial cytoplasm by the contraction of the tail region. This may be called viral infection of bacteria. It is not about the transfer of DNA into the host cell in the case of tailless viruses. Still, it is presumed that phage capsid breaks open, releasing the phage DNA into onto the bacterial cell wall. From there it gets into the cytoplasm by lysozyme activity. Following infection, the phage DNA takes command of the genetic mechanism of the host cell. During this, it acts as the master, and bacterial DNA serves as a slave. Master and slave hypothesis. So, in the bacterial DNA, loses its ability to replicate and transcribe. Eventually, it disintegrates into nucleotide units, which may be used for the replication and transcription of the phage DNA. Now, the phage DNA undergoes replication, transcription, and translation, making use of the molecular machinery of the host cell. During its replication, the phage DNA directs synthesis of a replication system to make identical copies of it. This is accomplished either by synthesis of phage-specific polymerase or by the phage-specific modification of bacterial polymerase. In both cases, several bacterial replication proteins may be employed. The transcription of phage DNA is almost always initiated by bacterial RNA polymerase. But after the first transcription event, either the bacterial polymerase is modified to recognize a phage promoter or a phage-specific RNA polymerase is synthesized. Transcription is regulated in such a way that phage proteins are synthesized as and when required. Usually there is a clear difference in the timing of the synthesis of phage-specific enzymes and structural coat proteins. This temporal difference is established by appropriately regulating the timing of transcription. Once the replication, transcription, and translation of the phage DNA are over, many hundred copies of it would be formed and sufficient amounts of phage proteins would be synthesized. The phage proteins include structural proteins, catalytic proteins, and maturation proteins. Structural proteins are the coat proteins which form the structural units of capsid. Catalytic proteins are the enzymes which mediate the assemblage of viral particles. Maturation process are the catalytic proteins which bring about the chemical modification and functional maturation of the phage DNA for packaging it with coat proteins. The coat proteins now assemble around each copy of phage DNA. This gives rise to a new progeny of mature phages. Shortly afterwards, the phage releases the enzyme lysozyme or endolysin. It recepts the bacterial cell wall and disintegrates or lyses the bacterial cell. This results in the liberation of the progeny phages to the surrounding medium. They may reinfect new bacterial cells to repeat the cycle. Some filamentous phages continuously release their progeny by unfolding the bacterial cell wall without causing serious damage to the bacterium. This process is called budding. In such cases, viral particles may be produced continuously over long periods of time. The successive events in the lytic cycle are regulated by three sets of viral genes, namely early genes, second genes, and late genes. Early genes constitute the first 20% of linear viral DNA. In about 4 or 8 minutes after infection, they undergo transcription with the help of RNA polymerase of the host cell and form viral mRNA. This mRNA soon gets cleaved into segments. Each segment undergoes translation on host ribosomes and produces a viral protein. Second genes constitute the next 20% of the viral DNA. They are of two major groups. The first group is concerned with the production of the enzymes which bring about the disintegration of bacterial DNA to provide free nucleotides for the synthesis of phage DNA. The second group is concerned with the production of the enzyme which bring about the actual synthesis of new phage DNA. Second genes undergo transcription and translation in about 6 to 15 minutes after infection. Late genes constitute the remaining 60% of phage DNA. They are concerned with the synthesis of capsid proteins and also with the packaging of phage DNA within capsid protein to form new phage. One of the late genes is especially involved in the synthesis of the viral enzyme lysozyme, which causes the lysis of bacterial cell. Late gene becomes active in about 5 minutes after the activity of the second gene is over. Viral infection of the bacterium is always accompanied by genetic recombination. In most cases, more than one strain of virus enter the bacterium. Since their genetic composition is different, there is every possibility for recombination. This viral recombination is particular in that the same DNA molecule can take part in many recombinations in the same infection cycle. The details of the molecular events of viral recombination are not quite clear, but still it is assumed that genetic recombination in viruses involve the formation of joins with gaps and overlaps. The gaps may be filled in by the activity of appropriate polymerase, and the overlap may be degraded by endonucleases. Sometimes members of the same strain of virus enter the bacterium. In such cases, a recombination takes place by breakage and heteroduplex formation. Since the genetic information in all the DNA is almost the same in this case, marked changes in the phenotypic manifestation of recombinant DNA may not be observed. Then part 2, like genetic transmission, lysogenic or temperate virus. More than 90% of phage is lysogenic. Among them, the lambda phage is the best studied one. In lysogenic phage, progeny viruses will not be produced and the host bacterium will survive and divide indefinitely for many generations. After that, the lysogenic cycle may come to a halt and lytic cycle may follow. During this, progeny phages are produced and then released by the lysis of the host cell. There are two types of lysogenic cycle. In the first step, the phage DNA gets closely incorporated with the bacterial DNA. This is the commonest type and is found in lambda phage. In the second phage type, the phage DNA behaves like a bacterial plasmid and it replicates. Independently inside the bacterium without getting incorporated with the bacterial DNA. This is less common or very rare and is found in the P1 stage, which infects E. coli. A bacterium which harbors a complete set of lysogenic and lytic phage gene is called a lysogen. The transformation of a normal bacterium to a lysogen by viral infection is called lysogenization. The process by which phage genome gets incorporated with bacterial genome is called insertion or integration. The close association between the viral and bacterial genomes leading to the repeated replication of viral DNA as part of the bacterial chromosome is called lysogeny. In phage DNA integrated with bacterial DNA is called prophage. The lysogenic cycle in which the phage DNA gets integrated with the bacterial DNA is completed in the following major steps. Like first, the phage at first adsorbs to the surface of the bacterium, then dissolves the bacterial cell wall by lysozyme action and finally injects its DNA to the bacterial cytoplasm. Soon after injection, the phage DNA undergoes transcription and translation inside the bacterium and produces a repressor protein and an integration enzyme called integrase. This is accomplished by utilizing the molecular machinery and ribosome of the bacterial cell. Very shortly afterwards, transcription is turned off by the repressor protein, and with the result that the phage genes get repressed. The phage DNA soon gets integrated with the specific site of the bacterial DNA with the help of integrase. This forms a DNA heteroduplex. In this integrated state, the phage DNA is called prophage. So during integration, the enzyme integrates recognizes the specific attachment site in the bacterial chromosome and binds with it. The host bacterium goes on growing and dividing indefinitely with the result that the prophage gets repeatedly replicated as a part of bacterial chromosome. Thus, the phage DNA is transmitted to several generations of host cell in a very short period of time. This harmonious association between the viral genome and bacterial genome is called lysogeny. Under certain conditions, the prophage gets extricated from the bacterial chromosome. This release of the phage DNA from the integrated state is called induction of the phage. The released phage DNA soon takes control of the genetic machinery of the host cell, follows a lytic cycle, and causes the lysis of the host cell. During this, it undergoes replication, transcription, and translation, producing multiple copies of viral DNA and coat proteins which get assembled to form progeny phages. So lysogenic bacteria have two characteristics, namely they can promote the induction of the prophage to initiate lytic cycle. They are insusceptible or immature to superinfection. They will not be reinfected by another phage which is similar or identical to the one which has already lysogenized it. Genetic transmission in tumor causing viruses. The infection cycle of a tumor causing virus such as the S40 virus which infects monkeys has two options, namely productive infection and abortive infection. Productive infection pathway is similar to the lytic cycle, then that it involves the production of viral progeny at the expense of the host cell. In abortive infection, the host cell is not lysed. In this case, the viral genome is believed to get integrated with the host cell genome as in lysogenic cycle. However, unlike in lysogenic phages, this integrated viral genome alters the genetic makeup and physiological functions of the host cell. As a result, the host cell undergoes a series of changes and becomes neoplastic or cancerous. The changes are collectively called transformation. In productive infection cycle, nearly 20% of the viral genome which constitute the early genes undergoes transcription inside the host nucleus and produces viral mRNAs. These mRNAs move to cytoplasm where they get translated to viral protein on the host cell ribosome. Of these proteins, the so-called T antigen, transformation antigen or tumor antigen is very important. It is essential for the replication of viral DNA, transcription of the late viral genes and activation of the enzymes of the host cell. Several hours later, replication of the viral DNA and transcription and translation of the late viral genes occur. As a result, many thousand copies of viral DNA and tens of thousands of viral protein molecules are formed. This get assembled into the nucleus of the host cell, forming progeny viruses. Shortly afterwards, the host cell dies. In abortive infection cycle, T antigen is synthesized by the activity of the early genes, but the late genes which code for capsid proteins and the enzymes of viral DNA synthesis are not expressed. Sometime later, the host cell undergoes repeated division and the resulting daughter cells show altered morphological and physiological features. This is called transformation. Some of the transformed cells may revert to their normal original form and become functional after a few days. So their transformation is called abortive transformation. The others stay on permanently in the altered state. This is called stable transformation. Next, genetic transmission in RNA viruses. In many RNA viruses such as measles virus, mumps virus, etc., genetic transmission and gene expression involves a direct flow of information from RNA to protein. In them, viral RNA acts as template and undergoes duplication as well as transcription. This duplication produces new genomic RNAs and transcription produces viral mRNAs. In both this process, the molecular machinery of the host cell is employed. Soon the viral mRNAs undergo translation on the host cell ribosome and produce specific proteins. Thus, in these processes, viruses information flow occurs from genomic RNA to mRNA to protein through gene expression. This is in conformity of the central dogma. According to the central dogma, during molecular transmission, genetic transmission and gene expression, information flows from DNA to mRNA to proteins through transcription and translation. Then genetic transmission in retroviruses, reverse transcription. We have a video about that. Retroviruses are RNA viruses with a single-stranded RNA, tumor viruses, human immunodeficiency virus, HIV, etc. In them, the course of the transmission and expression of genes is reversed, central dogma in the initial stage. In these viruses, information flow occurs from the RNA to protein by reverse transcription. Reverse transcription is accomplished by RNA-dependent DNA synthesis instead of the DNA-dependent RNA synthesis in usual transcription. In it, viral RNA acts as a template and guides the synthesis of DNA strand called single-stranded complementary DNA. with the help of the enzyme reverse transcriptase. This enzyme functions as a RNA-dependent DNA polymerase. The cDNA strands at first remains complexed with the RNA template, forming a RNA-DNA hybrid. Reverse transcriptase now changes its function and behaves as an RNase. This cuts away the RNA from the complex. Now the cDNA serves as a template and guides synthesis of a complementary companion strand. This forms a linear DNA duplex called DNA provirus. In this process, reverse transcriptase functions as DNA-dependent DNA polymerase. The linear DNA provirus is only an intermediate molecule in viral gene expression. Its own circularizes and gets integrated with the host cell DNA with the help of enzyme integrase. This integrated DNA provirus undergoes transcription as a part of the host cell DNA and produces viral mRNA and viral genomic RNA. Soon mRNA undergoes translation and produces viral proteins. This brings about viral gene expression. Viral genomic RNA gets packaged by viral proteins, forming retroviral particles. This in turn causes the transmission of viral genome to progeny viruses. Then reverse transcriptase is incapable of initiating the polymerization of ribonucleotides. So for its polymerase activity, it always requires a primer on to which DNA nucleotides can be incorporated. This primer is an oligonucleotide that brings a 3' OH group. It remains hydrogen bonded with the RNA template. The commonest primer is a tRNA fragment. So reverse transcription is significant in that it facilitates the retrieval of RNA sequence and the subsequent use as a source of genetic information. Also, it reveals that biological information in the form of RNA sequence can be converted to DNA sequence or vice versa. The molecular mechanism of the integration of provirus is not clearly understood. Once integrated, the provirus stays on in that condition and it is not usually extricated. Its presence does not inhibit the division of the host cell, so the daughter cells inherit the integrated provirus in the usual way. Then integration of the provirus may be accompanied by the rearrangement of sequences and the loss of some bases in the host cell DNA. However, coding sequences are never lost. The integrated DNA is the only source of progeny virus. There are several ways in which proviral integration leads to the transformation of the host cell. Some of them are the following. Integration occurring within the host cell inactivates the host gene. If the inactivated host gene is a regulator gene, regulation of cellular processes may get disrupted. Interactions isolates a host gene from its promoter and thereby prevents its expression. Integration causes a host gene to be influenced by viral promoter. This in turn leads to the overproduction of the host gene product. The product of viral gene expressed in the integrated provirus can directly or indirectly cause the transformation of the host cell. Integration often leads to chromosomal breakage and rearrangement of genes in the host cell. This in turn alters gene expression.